Hello everyone, and welcome to another recorded lecture of BIO101 online. Today we will discuss chapter 12, which is the central dogma of biology. So the early mid-20th century was a really exciting time for scientific discovery. And this started with an experiment done by Frederick Griffith in the 1920s. And he was investigating the virulence of Streptococcus, which is a type of bacteria that causes pneumonia. So he was studying this bacteria, and he was infecting mice with the bacteria. And there were two strains, specifically. There was an S strain that had a capsule on it, and there was a non-encapsulated strain that looked a little rough. So we call them the R strain. So we have two different strains, the S strain and the R strain. And interestingly, only when he injected the live S strain, which had a capsule, the mice died. But when he injected the R strain with no capsule, the mice were perfectly fine. Okay, nothing that special so far. But what was interesting is that when he killed the S strain with heat, it was unable to kill the mice. Right? So the heat allowed this bacteria to die, so it wasn't infectious. But if he injected the killed infectious bacteria plus the R strain, which was never normally infectious to begin with, together the mice died. Not only did the mice die, but he recovered live S strain from the dead mice. So maybe pause here and take a second to review the textbook of the explanation of this experiment and make sure you understand what he observed. But what he concluded was that virulence, the ability to infect, can be passed on from a dead bacterial strain to a non-virulent bacterial strain that's alive. So again, what he did was he took the killed S strain and mixed it with the live R strain, and that was enough for the mice to die. This means that something in the S strain, right, something in this, even though if it was killed, something in this S strain was able to pass on, right, the ability to infect, right? It was able to, so something was in there. And this is now called bacterial transformation, right? The bacteria were transformed, right? Griffith concluded that some substance necessary for the bacteria to produce a capsule and be virulent must have been passed on from the dead S strain to the living R strain, right? And the R strain bacteria were transformed, right? They were transformed and they were able to be infectious, right? Once they were mixed. So overall, right, the change in the phenotype of the R strain, right, going from non-infectious to infectious, must have been to a change in genotype. But what causes a change in genotype? Right? What is this material that's being passed from one strain of bacteria to the other? All right, so this is the next question. Griffith discovered this transforming substance. Something was transferred from one bacterial strain to the other and transformed the bacteria in the process. But what is this substance that's being transferred from one cell to the other? There were two candidates at the time. So DNA or proteins were both known to be parts of chromosomes, but we didn't know what is actually transmitting the information. Is it the DNA or the proteins? So proteins have 20 amino acids that can be sequenced in different ways. DNA and RNA only have 4 types of nucleotides. So people were saying, it might be more likely for proteins to be the candidate for hereditary material, because there are more letters in the alphabet. So there are a couple of requirements for whatever this hereditary material is. We have to be able to store genetic information in it. Right? We know that this material has information and it can be passed on from organism to organism. So it must be able to store genetic information. It must be stable and able to be replicated accurately during cell division. It also must be able to undergo mutations to provide genetic variability. And we know how important that is in sexual reproduction, for example. So these are the requirements for hereditary material. And researchers showed that DNA can fulfill all of these functions. So it was the group of many, many researchers in the mid-20th century that concluded that DNA is the hereditary material that stores genetic information. So in the mid-40s, this is 20 years after Griffith's experiment, Avery, McLeod, and McCarty did a very well-known experiment that demonstrated that DNA was the hereditary material. Originally, the scientists were saying, DNA didn't have enough letters. It wasn't variable enough to have genetic information. But it was this experiment that pretty much concluded DNA was definitely the hereditary material. What he did, so very similar to what Griffith did, he saw that when he took the R strain bacteria, and again, this is like pneumonia bacteria. So the R strain of pneumonia was unable to kill the mice. Only the S strain was. And when he heat treated the S strain, it was unable to be infectious. So that's very similar to what Griffith saw so far, right? And then what Griffith discovered was that if he added living R strain to heat-treated S strain, that the mice were able to be killed, right? This is what Griffith saw. This is Griffith's experiment, this top 4 part. Avery, McLeod, and McCarty added a little bit to it. What he did was he either added S strain treated with DNase, which is an enzyme that breaks down DNA, or an enzyme that breaks down proteins. So again, he took this bacteria, the heat-treated S, and he either destroyed all of their DNA or all of their protein. And he then mixed it with the living R strain and saw if it could infect the mice. He also tried this example with RNase, an enzyme that only destroyed the RNA. So by looking at this picture, what do you think he concluded? Right, and again, I'm going through this rather fast because I'm expecting you to have read the textbook already. This might be the second time you're watching this presentation. So you might have to pause and really think about the interpretation of the experiment. Why was the mouse able to live in this case, but not here? Right? The only enzyme that had an effect was the DNase that prevented the transformation from occurring, right? The only way that the mice still lived was if you destroyed all of the DNA of the S strain. Therefore, it must be that something in DNA, right? It's the DNA that is transforming, right? The DNA has the information that's being transferred between one strain to the other. And if you get rid of the DNA, you cannot transport information anymore, right? You can get rid of the protein and it's fine. You can still transmit DNA. You can get rid of RNA and it's fine. You can still transmit, the information. Right? So this showed that DNA was the transforming substance. So this is again just a more detailed look at it. You can see that he took the heat-killed S cells and he either destroyed all the protein, destroyed all the RNA, or destroyed all the DNA. And then he added the living R cells to each of the 3 tubes. And he saw that the only time that no transformation occurred was when the DNA was destroyed. So this concluded that transformation cannot occur unless DNA is present. Therefore, DNA must be the hereditary material. There's one more experiment you should be familiar with that again concluded that DNA is the hereditary material. And they used a virus called T-phage, and they labeled it radioactively in one of 2 different ways. They either labeled the DNA with radioactive dye, or radioactive isotope, I should say, or they labeled the protein of a bacteria. So what they did was They infected each type of virus, right? They used each type of virus to infect bacteria. And what they saw was that after they broke up, they dislodged all the viruses from the bacteria in a blender. They saw that what remained inside of the bacterium was radioactive DNA. The protein never made it inside the bacterium, right? No radioactive protein was ever inside in this strain. Only the DNA, only the radioactive DNA ended up inside the bacteria. So this again showed that viruses infect by transporting their DNA into cells, right? And the DNA is actually infectious. And DNA contains information that can be passed on from one organism to another. Right, so this is again saying DNA is the hereditary material, and that it was pretty overwhelming, right? You can't really argue too much about that. Right, so only DNA and not protein was the genetic material. And by the 1950s, we established that all organisms on this planet use DNA as the hereditary material, and that DNA, right, is a requirement for all living things, right? If you don't have DNA, you're not alive, pretty much. So this was the starting point for research in molecular biology, which is what we're going to talk about today. So let's go back to chapter 3, when we first introduced DNA. And we said DNA stands for deoxyribonucleic acid, right? So just like the name suggests, DNA is a nucleic acid, right? That's the type of biomolecule DNA belongs to. What are nucleic acids made out of? Right? If you recall, nucleic acids are made up of nucleotides, and every DNA nucleotide contains 3 things. What are those 3 things? Each nucleotide has a sugar. In the case of a DNA nucleotide, that sugar is deoxyribose. Every nucleotide has a phosphate. And every DNA nucleotide has a base. And the base can be one of 4 bases that all contain nitrogen. So we call those nitrogenous bases. And the base can be either adenine, we abbreviate that A, thymine for T, guanine is G, or cytosine is C. So all nucleotides of DNA can be one of 4 flavors, right? And they all look the same in terms of phosphate and sugar. The only way the nucleotides differ is in which of the 4 bases they have. Which is not part of a nucleotide? Right, the answer is B. Right, lipids are not parts of nucleotides. So nucleotides are polymerized in a DNA double helix, meaning when you look at a DNA molecule, it's a bunch of nucleotides attached together. Right, so a DNA molecule is a polymer, right, of many nucleotides. The nucleotides are the monomers. So we say that nucleotides join together to form strands. And DNA is always double-stranded. There are always 2 strands in a DNA molecule. Specifically, the phosphates and the sugars of each nucleotide form the DNA backbone. So as you can see here, there's a sugar, this is the sugar, and a phosphate. And these keep on repeating, right? Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, right? And each nucleotide contributes one of those phosphates and sugars. Here's like one nucleotide. The phosphates and the sugars form the backbone of each DNA strand. So we call those the backbone. So here you can see this red line is the sugar phosphate backbone. The bases from each DNA strand stick out. So just like a ladder, like the steps in the ladder, those are the bases. And the bases of each strand are joined by hydrogen bonds. Remember, hydrogen bonds are very weak. So the two strands of DNA are not actually attached. They're just attracted to each other by hydrogen bonds. And very, very importantly, base pairing is complementary, right? Not compliment with an I, you're not giving somebody a compliment, but complement, meaning that for every one letter, right, there is a corresponding letter. So for every G, it only knows how to pair with C. For every adenine, it only knows how to pair with thymine. So by knowing the sequence of one strand, you know the sequence of the other. And take a second right now to force this into your memory, that A pairs with T and C with G. All right, A with T, C with G. So keep on repeating that until it is stuck in your brain forever. So, like I mentioned, there are 4 nitrogenous bases that make up 4 different nucleotides. Adenine and guanine have 2 rings in their base. Again, these are just molecules of nitrogen and carbon, but they're double-ringed nitrogenous bases, and we call these purines. So adenine and guanine are purines. A trick to remember that is all the bases that end in -ine, like nine, are purines. Like guanine, adenine are purines. Thymine and cytosine nitrogenous bases only consist of 1 single ring, and those are called pyrimidines. So a purine can only match up with a pyrimidine. And Chargaff was a scientist in the 40s who discovered that the amount of adenine in the genome is equal to the amount of thymine, and the amount of guanine is equivalent to the amount of cytosine. So this is called Chargaff's rule. The exact percentages of each nucleotide will vary by the type of organism, right? But the amounts of A, T, G, and C in DNA, right, are all going to equal to 100%. They're always going to equal to 100%. And all of this suggested that DNA uses complementary base pairing to store genetic information. And just with 4 letters, this gives us an incredible amount of genetic variability. So if each human chromosome contains about 140 million base pairs, right? That's the amount of letters each chromosome has, right? You have 4 possible letters, A, T, C, and G, right? So it's 4 to the power of 140 million, which tells you how many different letters there could be on a chromosome. So this amount of variability explains how each species can still have its own unique base percentages. Which is not a DNA base? So the answer is C. Uracil is going to be an RNA base we learned about, but it's not found in DNA. So now let's talk about the DNA structure a little more. Each strand of a double helix has polarity, meaning each end is unequal, right? And this strand is oriented in a different direction as this strand. So let me explain what this means. Right? The end with the exposed phosphate is called the 5' end. The reason why it's called the 5' end is because the phosphate is attached to the 5th carbon in the sugar. So without going into too much detail, we start numbering the carbon here. It's 1, 2, 3, 4, 5. The phosphate is attached to the 5th carbon of the sugar. So we call the end with the phosphate sticking out the 5' end, or 5' end. Right? That's the 5' end. Here is the other 5' end. Notice how you can never have 2 5' next to each other. This will become very important. So there's one 5' here, another 5' here. Those are where the phosphates are exposed. The other end of the strand is called the 3' end. That's because the hydroxyl group the OH of the sugar, which you can't really see here, is attached to the third carbon of the deoxyribose. So there's an invisible OH here and up here. So there's the 3' end. The two DNA strands of a double helix are anti-parallel, right? Meaning that the sugar phosphate groups that are chained together to make each strain are oriented in opposite directions. One strand runs 5' to 3'. The other runs 3' to 5', right? 5 to 3, 3 to 5. However, genes are only read 5' to 3'. I'm going to repeat this a few times. This is really, really, really important, right? Information stored within DNA must always be read in the 5' to 3' direction. So what can this DNA sequence be? It can either be 5' AGTC or 5' GACT. Right? It can only be read 5' to 3' or 5' to 3'. Right? And you always have to indicate the 5' and the 3' ends on DNA. So take a second to try to tell me what the sequence of this looks like. What is this sequence? Right? You could write that it's 5' AGT ACT GA 3'. You can't just write letters. You have to indicate a capital letter and a period, just like you would in a sentence in English. Right? You have to indicate your punctuation, like 5' and 3'. So remember, DNA code has a direction. What type of bond holds the two DNA strands together? Right. The answer is D, hydrogen bonds. So Rosalind Franklin was not very famous at her time, but is now very well known as one of the most famous female scientists who was instrumental in discovering the double helix. A lot of people know about Watson and Crick. Not as many people know about Rosalind Franklin, because without Rosalind Franklin, Watson and Crick would have never had their discovery. And Rosalind Franklin was Jewish, and she fought a lot of obstacles to try to get her work published as well. So she should be actually celebrating her 100th anniversary in the year 2020. She would be 100 years old. But unfortunately, she died at the young age of 37. So Rosalind Franklin studied coal in her as her career, but she also studied the structure of DNA. And she used X-rays in order to study the structure of DNA. So it's something called X-ray crystallography. And X-ray crystallography is how we study the structure of proteins today. This was a new field in Franklin's time, right? Because this is, again, we're talking about like the 40s. So what she did was she was able to crystallize DNA and used X-rays to bombard the crystalline DNA, and she was able to produce X-ray diffraction photographs. Again, very tricky to kind of explain, but when DNA is in a certain form, it can be crystallized, and that means you can bombard it with X-rays, and the X-rays will bounce off of different atoms in DNA and create a pattern on like a film, like X-ray film. And that can help us deduce the structure of this crystallized molecule. So she provided evidence using X-ray crystallography that DNA had the following features. She proved that DNA is a helix and that some portion of the helix is repeated over and over again. So a colleague of hers, Wilkins, showed her photo to James Watson, who automatically recognized what this photograph meant. And he was able to tie that in with his work So with Watson and Crick, right, together, modeled the double helical 3D structure of DNA in the 50s. So Crick was actually English and Watson was American, but together, using Franklin's X-ray diffraction data, they proposed a model of DNA in 1953, and they received the Nobel Prize in 1962. So Watson and Crick together determined that the two strands run anti-parallel to each other, right? One has to go 5' to 3', the other goes 3' to 5', right? They said that the bases must form the horizontal rungs of the ladder, so to speak. Like the steps of the ladder are the complementary bases, right? Where the sugars and phosphates form the backbone of the DNA. So this conclusion was kind of the final piece of the puzzle to figuring out the structure of DNA. And DNA is most stable when it's in its double helical form. And the structure of the double helix is also due to hydrogen bonding. So hydrogen bonds keep the two strands together, but it also keeps the double helix in its intact shape. And we can think about just how stable DNA is, because you can sequence the DNA of a mummy in ancient Egypt from thousands and thousands of years ago and still see functional genes. Not functional like they're not really producing protein, but you can still read the sequence of some of these genes. So DNA must be very, 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 very stable, right? If it can not only remain for thousands of years, but it could also remain in the heat of Egypt, for example. So DNA is very stable. Here's a quick summary slide of the major discoverers of different aspects of DNA and then what their major contribution was. So I'm not going to test you on this, like specifically who discovered this, but I think it's very important to acknowledge how the story of DNA was uncovered. So let's watch a video on DNA together.
The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides, which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands.
Great. So let's review. DNA and RNA belong to the macromolecule group known as what? Nucleic acids. The monomers of DNA and RNA are known as what? Nucleotides. How many different nucleotides are there that make up DNA? 4. And what are the base pairing rules? Adenine goes with thymine, cytosine goes with guanine. Great. So let's quickly recap some information about genes. So a gene contains biological information, usually instructions to make a specific protein. And these instructions, these genes, are spread out in long DNA molecules called chromosomes. We have about 20 to 25,000 different genes that code for proteins that are divided amongst 23 chromosome pairs, right? We have 46 chromosomes. 23 came from mom, 23 came from dad. And all of those chromosomes together make up the genome. So the genome, you could think of it as like a user's manual or like a cookbook. And the genome, like a cookbook, has a bunch of different recipes on how to make different proteins. Where is the genome stored? So if in prokaryotes, the DNA is stored in the cytoplasm, and they have a single circular chromosome. It's located in a nucleoid region of the bacterial cell, but it's not very complex. In eukaryotes, we have a nucleus that protects DNA, and eukaryotes have multiple linear chromosomes made of chromatin. All right, so DNA gets wrapped up into chromosomes, and it's found in the nucleus of eukaryotic cells. All right, and for example, in humans, we have linear chromosomes. All right, we have 23 pairs of linear chromosomes. So you can pause here, and the answer is B, the nucleus. Where is the E. coli genome contained? The answer is A, the cytoplasm. So the Human Genome Project was able to sequence all of the DNA in our genome. And the information was really, really important because we could discover a lot of secrets of DNA and also understand which genes are associated with disease. So we know that each cell contains 46 chromosomes. If we were to unwrap the chromosomes, we would see a DNA double helix. The DNA double helix consists of a sugar phosphate backbone and letters, which represent the nitrogenous bases A, T, C, and G. All right. Together, the DNA in the chromosomes, which are more than 3 billion letters, make up the human genome. There are, we said no more than 30,000 genes. So it's about 20 to 30,000 genes, which are instructions to make proteins. And the proteins are the physical hardware that make up our cells, our organs, etc. They do the chemical reactions, transport materials. There are some mutations that are like typos in the instructions, and that can cause disease. And we were able to discover a lot of different genes that lead to disease when mutated, such as cystic fibrosis or sickle cell anemia or Huntington's disease. This is just a little fun analogy for you to understand just how large your genome is. Each cell of your body has 2 meters of DNA. That's 6 feet of DNA in one microscopic cell. It's already kind of impossible to imagine. The human body has between 10 to 40 trillion cells. So if you were to take all of your cells and open up the nucleus and unwrap all the DNA and line it up end to end, that would give you about 20 billion kilometers of DNA. Your DNA can stretch to the sun and back over 60 times. So you could go from the Earth to the sun and back over 60 times. And that will still not be the entire length of your genome. Just to compare, right? So we're going from here to the sun and back 60 times. That's how long the genome is of one human being. If the DNA in a single cell were printed in size 12 font, Times New Roman, and the pages were all stacked up, how high would the pile be? Well, again, if you just typed up your letters of your genome in a single cell, that stack of paper would be about 425 feet high, which is about midway between the Washington Monument and the Statue of Liberty. So that is a lot of letters, right? So it's 3 billion letters of DNA in each cell. Well, actually, you could say more. That's half. So it's 3 billion base pairs are each cell. So how can all that DNA fit inside of a tiny cell? Right? So imagine that the DNA is like loose thread and it's kind of like all over the place. But what we can do is we can wrap it around spools to make it a lot more organized and contained. So those spools are called histones. And histone proteins are actually octamers, meaning they have 8 parts to them. They're 8 subunits. And these act as protein spools to wrap DNA around. And together, right, DNA wrapped around histones is called chromatin. Right? And that's what chromosomes are made of. Chromosomes are made of chromatin. So here's our DNA double helix, and it gets wrapped around histones like beads on a string. One bead with string wrapped around it, meaning one histone with DNA wrapped around it, is called a nucleosome. And DNA is 10,000 times more compact when it's wrapped around histones. So the DNA gets wrapped around histones like beads on a string, but then these continue to compact and compact and compact until you can basically get a mitotic chromosome, which is as condensed as you can be. And the DNA molecule is now 10,000 times shorter than its original length. So what is chromatin made out of? You can pause here. And the answer is D, DNA and histone proteins. Which is the largest? You can pause here. And the answer is a chromosome. Which is the smallest? Pause here. And the answer is a nucleotide. So now we will discuss DNA replication. So DNA replication is only going to happen right before a cell divides. That's the most important thing to understand right now. DNA replication is only going to happen in cells in which phase? Right? It's interphase, and specifically which part of interphase? It's S phase. Right? So in cell division, a parental cell divides into 2 daughter cells, but for each daughter cell from the division to have identical DNA, the parent cell must first replicate its genome. Right, so we said this happens in synthesis phase of interphase. So in prokaryotes, this is really, really simple. Bacteria have a single circular loop of DNA. There's a certain location called the origin of replication, and replication just moves around the circle in both directions. So try to visualize. You could start at this origin, and the DNA strands can separate, and you can build new DNA in both directions. We therefore get 2 identical circular chromosomes, and this takes about 40 minutes. Interestingly, the cell only takes 20 minutes to divide, so a new round of replication could begin before the previous round is even completed. So you could have double the replication happening. So it's very quick, so it's relatively simple compared to eukaryotic. So what I'm going to show you now is what eukaryotic DNA replication looks like. This is an overview, so it's not going to give you, the details, but this is what we're going to take a look at in the upcoming slides.
DNA replication is the process in which one double-stranded DNA molecule is used to create 2 double-stranded molecules with identical DNA sequences. For this process to occur, the 2 DNA strands are separated, and the DNA replication machinery is assembled at the replication fork. The 2 DNA strands are used as template strands to create newly synthesized DNA strands. The key feature that allows a DNA molecule to produce 2 DNA molecules with the same DNA sequence is the ATGC rule, also known as Chargaff's rule. According to this rule, adenine in one strand only hydrogen bonds with thymine, and guanine only hydrogen bonds with cytosine. For example, an incoming nucleotide with a cytosine base will only hydrogen bond with a guanine base in the template strand. By this mechanism, one double-stranded DNA molecule provides the information to create two double-stranded molecules with identical base sequences. Each copy contains one newly synthesized strand and one original strand. This mechanism is called semi-conservative DNA replication because half of the DNA molecule is conserved from the original molecule.
So now we're going to zoom into what we just saw. So we said the first thing that has to happen is the DNA strands have to unzip. Right? The first thing is they have to separate. And the hydrogen bonds are what are keeping the two strands together. So a special enzyme called DNA helicase comes in and it starts breaking up the hydrogen bonds between the double helix. And this happens at numerous points along each linear chromosome. So replication in eukaryotes, it's still not as fast as prokaryotes, but it can still enhance the time because there are multiple origins of replication. And these hydrogen bonds are really crucial because it allows the DNA double helix to open, like separate temporarily, and then reform a new helix as new nucleotides are joined in. So once the DNA double helix, this is called the parental strand in blue. This is the original DNA molecule we're about to copy. So this is the parental strand in blue, and this is going to unwind with the help of DNA helicase. And now each parental strand can be used as a template for a new strand. What that means is a special enzyme called DNA polymerase is going to add in complementary nucleotides. If this original DNA molecule had an A, that means only a thymine, a T nucleotide, can go on this new strand, right? And so forth. So then if you see, and then a C, what can go with a C? It can only be a G over here. So at the end of DNA replication, you have two identical double helices, but half of each strand is newly formed. Like the one in gold was all brand new. That was consisting of new nucleotides. But half of each strand is still conserved from the original parent molecule. So each of these two strands are identical to this original strand, and they're identical to each other. But it's called semi-conservative replication because half of the strand is from the original parent and half of the strand is new nucleotides. Right, so it's semi-conservative replication because each daughter DNA double helix contains an old strand from the parental DNA double helix and a new strand built of brand new nucleotides from DNA polymerase. Right, so again, just to illustrate semi-conservative replication, each strand separates and serves as a template, like a mold, for the new daughter strand to be built in. So if there's a T in the parent DNA, an A will be put in. If there's a G, there's going to be a C. G, another C. And these new nucleotides will be put in one by one. All 46 chromosomes get replicated at the same time in the case of humans. And again, these chromosomes get opened up along different points. Right, so each chromosome can be opened up along different points. So it happens rather quickly in a matter of hours. Right, so if we start with this DNA sequence 5' to 3', look how they're anti-parallel to each other. And these dotted lines represent the hydrogen bonds. We're going to separate the blue parents, and new green nucleotides will be filled in. And what we get is two identical double helices, right? So we started with one, and now we got two. And it's semi-conservative replication. The image on the right shows the DNA before replication. Which of these shows or best represents the newly created red strands of DNA after replication is complete? So the new strand, the new daughter molecule, will look like C, right? It will consist of half of the original and half of new nucleotides. So the enzyme that makes DNA is called DNA polymerase, right? DNA is a polymer of nucleotides. So the enzyme that makes the polymer of nucleotides is called DNA polymerase. Right? So DNA polymerase basically attaches complementary DNA nucleotides together according to the template DNA sequence. Interestingly and very importantly, DNA polymerase can only add nucleotides to the 3' end of another nucleotide. Think about that. So there's only one direction that DNA polymerase can go in. It can only add nucleotides to the 3' end of another nucleotide. Meaning, the 5' phosphate of an incoming nucleotide, of a new nucleotide, has to be joined to the 3' hydroxyl group of the preceding nucleotide. So what direction is DNA made in? Is it 3' to 5' or 5' to 3'? New DNA must be made 5' to 3' if new nucleotides can only be added to the 3' end. Right? Only 5' to 3'. So by looking at this is like zooming into the middle of a gene. Name the 5' and 3' ends. Are A, B, and C 5' or 3'? And how do you know? So pause here and try to figure that out. So this lower strand, this is the template strand, right? And the new strand is being added one by one, and this was the most recent nucleotide added. Since this is added most recently, we know this has to be the 3' end over here. This is the most recently added nucleotide. This must be the 3' end because this is where the replication is going. If this is the 3' end, that means this must be the 5' end, right? A must be 5', and that means B must be 3'. And if B is 3', that means, what is C? C is also 5'. So if that's not clear, I suggest pausing here, going back to the slide where I introduced that DNA is anti-parallel and the strands are polar, and make sure you understand what 5' and 3' really mean. Right? So this is the 3' end, another nucleotide will come in, and the phosphate of this new nucleotide will attach to here. And it will keep on going in this direction, from 5' to 3'. So I'll draw it on here just to make it as clear as possible. Right, this 5', that's 3', 3'. Prime, 5'. And remember, you always want to make sure that DNA is anti-parallel. You can never have two 5' on top of each other or two 3' on top of each other. It must be anti-parallel. What enzyme connects nucleotides? The answer is C, DNA polymerase. In which direction does DNA polymerase add new nucleotides? The answer is only A. Where on the parental or the template strand does DNA polymerase start replication? So pause here and think about this for a second. So if it makes DNA 5' to 3', it has to start at the 3' end of the parental DNA template. So then it can make DNA from 5' to 3'. And this will be very clear, hopefully, in the next couple of slides. So let's talk about this step by step. The first thing that has to happen is DNA helicase breaks the hydrogen bonds between the two strands of DNA of the parental strand. And right, the two complementary strands are now separate from each other. But they're very likely to just want to reform. right? Because they're kind of attracted to each other. So other proteins called SSB proteins, they stand for single strand binding proteins, kind of act like clamps. They clamp onto each single strand and prevent them from reforming hydrogen bonds and forming the double helix again. So helicase and SSB together make sure that the DNA strands are separate from each other so replication could occur. And what I want you to notice here is that helicase creates a fork in the road. This is called a replication fork because now these are split. So just keep this in mind. And of course, in reality, this is not going to be the end of a chromosome. We're in the middle of a chromosome. So there would be another helicase going in this direction too, but we'll hold that for a second. This is where I want to introduce the whole idea of how helicase unzips DNA at several points along a chromosome. So let's say this is one chromosome that gets replicated. It's not like DNA helicase starts at one end and just unzips in one direction. It's not that simple. What happens is that there are multiple locations within the chromosome that are called origins of replication, and this is where helicase starts. So a helicase will start opening up here, will start opening up here, and will start opening up here. And what this does is create replication bubbles, these little openings, little bubbles. Ignore the blue for now. And at each end of the bubble, we have a replication fork. Right? This is where the two strands separate from each other. So we have replication forks at each end of the replication bubble. And what we're going to see is that DNA, the new DNA in blue, gets made as the parental DNA separates. So try to visualize that as these purple DNA strands separate, the new blue strands get made more and more until they all converge and you get two daughter DNA molecules. Right, so try to visualize how replication bubbles kind of continue to open up wider and wider until they just form two separate identical daughter DNA molecules. So let's zoom into one of these replication forks. So we're zooming into just one of the replication forks, one of the origins of replication. And of course, I'm notating 5' to 3'. So here is my little helicase enzyme, and it's going to fly over to the first origin of replication, and it's going to open up the double helix by separating the hydrogen bonds. Great. So now we have no more hydrogen bonds between these two parental DNA molecules. This is the replication fork. So helicase is going this way. It's going to move this way. Meanwhile, there's another helicase in the other direction moving this way. Right? So one helicase will open this way, another helicase will open in the other direction. Right? Notice that, I'm going to go back for a second. Right? One helicase is opening this way, another helicase is opening this way. Here's another helicase, another helicase. So helicases are just attached to every origin and start opening DNA in both directions, just breaking hydrogen bonds. So we're breaking hydrogen bonds, and now we have DNA polymerase come in. And DNA polymerase is the enzyme that can actually build nucleotides. It can read the template sequence, right? The A's, T's, C's, and G's of the template, and then it can build in the corresponding strand that's identical. But where does DNA polymerase want to start? So where is DNA polymerase going to start? Right, we said DNA polymerase wants to start toward the 3' end of the template DNA, right? Because it can make DNA, it can only join nucleotides 5' to 3', right? So the DNA polymerase must bind to the 3' end first, and then it can make new DNA 5' to 3'. But what's the issue, right? How could DNA polymerase, it can't bind here at this 3' end because it's sealed. And it can't bind to a 5' end because then it would make DNA 5' to 3', and that would be parallel. You can't have DNA polymerase start here and make 5' to 3' DNA because that would be parallel. So we have an issue here. The first part is pretty easy. We can make one strand very simply. So the leading strand is the strand that is built continuously in the 5' to 3' direction. So at this replication fork, we have DNA polymerase starting toward the 3' end, and it will start making new DNA 5' to 3' continuously in the same direction as helicase is unzipping. So helicase is going this way, it's going from right to left, it's unzipping, unzipping, and DNA polymerase can start at the 3' end of the template and start making new DNA 5' to 3'. This is an easy strand to make continuously. So this is made 5' to 3'. DNA polymerase is basically following helicase, building new DNA. However, we can't do that on the lower strand, right? We can't just have DNA polymerase make 5' to 3' DNA. The leading strand is built continuously because DNA polymerase is going in the same direction as helicase. That's not the same situation as what we have for the lower strand. So the leading strand is built continuously in the 5' to 3' direction. We call that leading strand synthesis. Right? It's built continuously because it's the same direction as helicase. So the lagging strand cannot be synthesized continuously because DNA polymerase has to wait for helicase to unzip the template ahead of it. So let's try to visualize this. Right, we said the top strand can be easily made because helicase is unzipping this way, so polymerase can just follow it along, making new DNA 5' to 3'. That's not the case down here. Right, we call this the lagging strand because DNA first has to be unzipped, and then polymerase can come in and start making new DNA 5' to 3'. Then it has to unzip a little more, and then it can make DNA 5' to 3'. So it makes DNA in these short little fragments called Okazaki fragments. So Okazaki fragments are short fragments that could be laid down as soon as a little bit of the template opens. So I'm going to try to explain that. I'm going to show you in an animation a lot more easily. But this was the first Okazaki fragment to be put down. Right? When helicase just started opening up the template at the beginning, one DNA polymerase can start up here, making new DNA 5' to 3' continuously. But this one down here had to wait until DNA opened a little bit, so then DNA polymerase can come back here and start making a little fragment 5' to 3'. So the first fragment that was made was 5' to 3', was this fragment. Then DNA had to open up even more, and then this fragment could have been put down. But now polymerase still has to wait, and then DNA was opened up more down here, and then this can be put down. And then this can be put down. And now we have to wait for this to unzip this little portion. Once this unzips there, then we can put that down. And then we can put this down, and so forth. So we're almost going in reverse in lagging strand synthesis. Right? It's almost like backstitching in a way. If you're used to sewing, you're going like back and forth, and then back and forth, and then back and forth, back and forth, back. So these get made in fragments discontinuously because polymerase cannot go in the same direction as helicase. Right? It's going to only make new fragments 5' to 3'. Which means it has to wait a little bit. So that's why it lags behind the other strand. That's why it's called lagging strand. DNA polymerase is unable to replicate the ends of chromosomes because of lagging strand synthesis. It could never really get to there's going to be a point where it can't open up anymore. It can't really grab onto anything and start making new DNA. That's the short of it. So the ends of chromosomes have to be replicated by an enzyme called telomerase. And telomerase, we said, is upregulated. It's overexpressed in cancer cells and stem cells. And it's now known that the longer the telomeres are in your cells, the longer they can replicate. Right? So every time a cell divides, telomeres get shorter and shorter and shorter, and that kind of limits the life of a cell. But in cancer cells and stem cells, there's this overactive enzyme telomerase that continually adds to the telomeres. And that makes them live infinitely, theoretically. So how do those fragments get joined? Right, so DNA ligase is an enzyme. DNA ligase is a special enzyme. It's almost like DNA glue that glues together the Okazaki fragments to make one nice sealed daughter strand. And just one more thing I didn't mention. DNA polymerase can't just start copying DNA on its own. It requires a little primer to start. And the primer, it's like a little kickstart. It tells DNA polymerase where to start replicating. The primer is actually made of RNA instead of DNA. And that's shown in green here. This is a little RNA primer that was put down by an enzyme called primase. So primase comes in, puts down a little primer, and that primer is made of RNA. And then DNA polymerase will recognize this and start branching off from that primer, making new DNA 5' to 3'. The RNA nucleotides in the primer are later replaced with DNA nucleotides. So then we have a 100% DNA chromosome. There's no RNA in the chromosome afterward. But just remember, DNA polymerase can't just start on its own. It needs to be told where to start replication by a little primer. And primase is an enzyme that puts little primers at the origin of replication, so DNA polymerase knows where to go. So this is an overview of everything we spoke about so far. DNA helicase started over here and is unzipping, right? It's unzipping DNA this way, going from left to right. So it created this replication fork, right? This is called a replication fork over here. And of course, single-strand binding proteins are going to help hold the two strands apart. Right, so here's helicase, creating a replication fork. SSB proteins will hold the two strands apart. And now we can see, since helicase is going this way, right, one strand could be made continuously. So we know this strand is made, this is the leading strand. This could be made continuously, 5' to 3'. However, this strand cannot be made continuously because helicase is going in the opposite direction of polymerase. So again, what has to happen is there's a short RNA primer, right, and then an Okazaki fragment. Short RNA primer, Okazaki fragment. RNA primer, Okazaki fragment. And at the end, DNA ligase will come in and seal up all the spaces in between the fragments to make one continuous molecule. And at the end, we're going to have two separate double helices that look exactly like the parent. So again, DNA helicase unzips. This is the replication fork. One strand can be made continuously, right? This 3' end of the parental strand is exposed, which means this will be the leading strand synthesis, right? The strand with the 3' end is where DNA polymerase is going to start, making new DNA 5' to 3'. So it's going to go in the same direction as the helicase is opening up. On the other hand, the lagging strand was synthesized discontinuously, right? And primase makes a short primer. It's not shown up here, but it happens here too. Over here, there's a short primer put in first. And then it's extended by DNA polymerase to form an Okazaki fragment. So this was the first one built, 5' to 3', then this one, then this one, then this one, and now then this one's going to be made, and then that one. But meanwhile, this top strand will be made continuously 5' to 3'. I hope that makes sense, at least a little bit. And this replication is very accurate. There is a mistake made once per 100,000 base pairs, but we have proofreading capabilities that allows polymerase to kind of Control Z and cut out something. It can undo a mistake it made, and it can cut it out and be corrected. Why do you think that's important? Why is it important for proofreading capabilities? We don't want too many mutations to accumulate, especially if it's in a crucial gene. But yet, mutations are important for diversity. So there's a fine balance. Like, some errors are necessary, but it has to be the right balance of errors and accuracy. Right? What would happen if DNA polymerase couldn't proofread? You get tons of mutations, right? But what if DNA polymerase were 100% accurate all the time? Right? Then you would get no diversity. So to summarize, we started with helicase unzipping. The double helix, SSB proteins prevented them from reforming. Primase starts by putting a primer on. Then DNA polymerase recognizes that primer and begins DNA synthesis. There are two strands that are replicated differently, leading and lagging. DNA ligase then seals together the Okazaki fragments, and we have two identical daughter molecules at the end. So pause here and name each enzyme as I list the description. All right, so pause here. What unzips the DNA double helix? Helicase. What copies DNA? Polymerase. What attaches the fragments together? Ligase. What makes the RNA primer? Primase. Which direction is helicase going in this case? Is it going up or down? Well, it's going up. It started down here and it's opening this way. Again, make sure you can understand 5' and 3'. If I show you a picture, you should be able to draw just by showing these arrows, knowing that this strand is made continuously. You would know that this is the 5' end, and this is the 3' end, and this is the 3' end, and this is the 5' end. Right, so make sure you can label something like this. So here, I'm not going to show this whole video, but this is included in your ebook. This is a really interesting talk. I actually want you to understand how replication occurs in real time. So this is a good, quick animation that shows you DNA replication in accurate speed.
Let's begin. This is DNA in its classic double helix form, and it's from X-ray crystallography, so it's an accurate model of DNA. If we unwind the double helix and unzip the two strands, you see these things that look like teeth. Those are the letters of genetic code, the 25,000 genes you've got written in your DNA. This is what they typically talk about, the genetic code. This is what they're talking about. But I want to talk about a different aspect of DNA science, and that is the physical nature of DNA. And it's these two strands that run in opposite directions for reasons I can't go into right now, but they physically run in opposite directions, which creates a number of complications for your living cells, as you're about to see, most particularly when DNA is being copied. And so what I'm about to show you is an accurate representation of the actual DNA replication machine that's occurring right now inside your body, at least 2002 biology. So DNA is entering the production line from the left-hand side, and it hits this collection, this miniature biochemical machines that are pulling apart the DNA strand and making an exact copy. So DNA comes in and...
So just to note, this is helicase. That's unzipping the DNA into two strands. This is the parent DNA. This is helicase over here. So it's unzipping, and this is the replication fork.
It's this blue donut-shaped structure, and it's ripped apart into its two strands. One strand can be copied directly, and it can be seen spooling off down to the bottom there.
Right,
but things aren't so simple for the other strand because it must be copied backwards.
Right, so just look back. This is continuous.
Spooling off down to the bottom there. Chemical machines that are pulling apart the DNA strand.
Oops, sorry.
Really began. It's this collection, this miniature biochemical machines that are pulling apart the DNA strand and making an exact copy. So DNA comes in and hits this blue donut-shaped structure, and it's ripped apart into its two strands. One strand can be copied directly and it can be seen spooling off down to the bottom there, but things aren't so simple for the other strand because it must be copied backwards. So it's thrown out repeatedly in these loops and copied one section at a time, creating two new DNA molecules. Now, you have billions of this machine right now whirring away inside you, copying your DNA with exquisite fidelity. It's an accurate representation, and it's pretty much at the correct speed for what is occurring inside you. But I've left out error correction and a bunch of other things. This was work from a number of years ago. Thank you.
So that's pretty interesting, and that's happening inside of all your cells right now. So now we will start talking about RNA and the central dogma of biology. So the whole time we're talking about DNA, but what about RNA? So RNA and DNA are close chemical cousins, but they have some key differences. The sugar is a big difference. So there's deoxyribose in a DNA nucleotide, but ribose in an RNA nucleotide. That difference is because there's one less oxygen in DNA. Right? That's what's called deoxy. There's one less oxygen, whereas in RNA there is an oxygen here. There are 4 bases, A, G, C, and T in DNA, but there's no such thing as T in RNA. No such thing as thymine in RNA. Instead, there's a uracil. So adenine pairs with a uracil in RNA. There's no such thing as thymine in RNA. DNA is double-stranded. RNA is generally single-stranded, but it can form some really complex secondary structures, as we call them. But generally, it's single-stranded, where DNA is a double helix. DNA stores protein encoding information and transmits it from generation to generation. RNA also carries the information to make proteins, but it's what actually can go to the ribosome. So that's what we're going to talk about. RNA can go to the ribosome to make proteins. DNA cannot. DNA is stuck in the nucleus. So that's what we'll be talking about in this next few minutes. And remember that RNA can also have catalytic activity. So some RNA can form ribozymes, which can actually act like an enzyme. And it's made of RNA nucleotides, not amino acids. So the central dogma of biology sounds kind of like, I don't know, it sounds very profound and deep. And it kind of is, because it was a huge breakthrough at the time. And this was pioneered by Crick, of Watson and Crick, after they discovered the double helix, quote unquote discovered, right? They got the recognition for it, at least. And what they established was that the genetic code is made up of DNA, and that contains instructions to make proteins. Right? This is the central dogma, is that we have DNA that gets passed from generation to generation, and those genes have instructions to make proteins. Those DNA instructions get rewritten into RNA first, and then ribosomes can make proteins using the RNA message. So the central dogma is sometimes abbreviated as DNA to RNA to protein, like DNA arrow, RNA arrow to protein. It's a good way to memorize it. There are some issues. It's a little more nuanced than that, as we now know. But the general idea is that DNA gets rewritten, gets transcribed into RNA, and then RNA gets translated into proteins. So this is called gene expression. Right? The central dogma of biology explains the mechanism of gene expression. When a gene is expressed, that means the information is made into a protein. So this is a little conceptual, but I want you to really understand this. So the central dogma of biology says that the DNA in genes specifies information, but that information is not structural or functional. It's digital, meaning it's consisting of A's, T's, C's, and G's. It's two-dimensional. Just like, and might be a little too complicated, like software can be coded in binary digits, like zeros and ones. DNA is similar in that way, and it uses A's, T's, C's, and G's as its language. The genetic information is expressed into structure and function through proteins. So the information in DNA is like the software that codes for the hardware of protein, and the proteins are what do the actual work of the cell. So the expression of genetic information into structure and function is explained by the central dogma. Specifically, the DNA code of a gene determines the sequence of nucleotides in an RNA, and the RNA dictates the primary structure of a protein. Remember, the primary structure of a protein is just the sequence of amino acids. And we know that the sequence of amino acids will determine how the protein folds, right? So the protein folds into a very specific structure. And we know in biology that structure determines function. So let's try to tie all this together. So DNA has instructions on how to assemble different building blocks, amino acids, together based on the chemical properties of those amino acids and the sequence that they're in. They will fold in such a way that will give them a very particular structure. That structure of the protein will determine how it can function. And that's, again, structure determines function. And that's what the central dogma explains. So to make proteins, to have the hardware, we need two stages, right? There's two phases of gene expression to make proteins. The first is called transcription. Transcription is how DNA gets rewritten. It gets transcribed into RNA. This occurs in the nucleus. We then have translation. Translation is when proteins are made on ribosomes using the instructions from RNA. So the central dogma explains how transcription rewrites DNA into RNA, and then ribosomes use the information in the RNA and translate it into proteins. You could think about the two stages of protein production like making brownies in a way. So imagine you have a cookbook that's like a chromosome with multiple genes, right? Your cookbook has like hundreds of different recipes in it. And chromosomes have thousands of genes on them. But we don't want to make every single recipe at once, right? We only want to make the recipes that are necessary for now. So if I'm having a party and I want to make brownies, I don't want to make a salad right now. I don't want to make everything and soups and chicken and beef. I don't want to make everything in the cookbook. I just want to make brownies. So suppose this cookbook is very heavy and it's stuck in my library. I don't want to carry the whole cookbook down. I'm just going to make a copy of the brownie recipe, right? Why carry this whole book? So what I'm going to do is just copy down. I'll make a photocopy of just the brownie recipe. That's like transcription, right? Instead of taking the whole book and making the whole book, we're just going to copy what we want. That's transcription. Translation is when we assemble the ingredients, like amino acids, together in a very specific order and sequence according to the recipe. Right? That's what translation is doing. Translation is using the recipe contained in the mRNA, in the RNA code, to assemble amino acids in such a way to make a functional protein. So again, make sure you conceptually understand this before moving forward. So this is what the central dogma of biology says, right? DNA gets replicated and repaired. So DNA can kind of do its own replication. But DNA gets transcribed into RNA in the process of transcription. RNA can also be made into more RNA. So RNA can replicate itself into more RNA, but that's a separate story. But the RNA gets translated into proteins, right? And that happens on ribosomes in the cytoplasm. There are exceptions because we now know that certain bacteria have enzymes, and certain viruses have enzymes, rather, that convert RNA to DNA. So there's something called reverse transcriptase, which is an enzyme that converts RNA to DNA because it does the reverse of transcription. It's the reverse transcriptase. So the central dogma is not just DNA to RNA to protein. It's a little more complicated than that. But here is a good video that we're going to start to introduce us to the central dogma of biology.
The sequence of nucleotide bases in DNA carries genetic information in units that are called genes. Structural genes encode the information for specific proteins. To create a protein, a gene must first be transcribed into a sequence of nucleotide bases in a messenger RNA molecule. The messenger RNA sequence is then translated into an amino acid sequence of a protein. This sequence of amino acids in a protein molecule determines the shape and chemical characteristics of the protein. Thus, each structural gene specifies a specific protein in the cell that carries out a specific function based on its chemical characteristics and molecular shape. This function of the specific protein gives the cell and the organism the specific trait coded by the gene.
So pause here and try to answer this rapid response question. So, the main function of DNA is to encode proteins, right? That's the main function. I don't know why that came up. But the main function of DNA is just to encode proteins. It has the instructions to encode proteins, but the cell needs to function. So now let's talk about transcription in more detail. So, what's the point of transcription? We said already that ribosomes in the cytoplasm, right? Ribosomes in the cytoplasm or on the endoplasmic reticulum make proteins. Ribosomes connect amino acids to make proteins. DNA has the instructions to make those proteins, but DNA is stuck in the nucleus. DNA cannot leave the nucleus. However, RNA is a close chemical cousin of DNA that can leave the nucleus. So it can almost act like a messenger between the DNA in the nucleus and the ribosomes in the cytoplasm. So what happens is DNA gets transcribed, gets rewritten into RNA so that the genetic message can be sent to the ribosomes in the cytoplasm. And therefore, the proteins can be made using the instructions originally contained in the DNA. And before we had electronic transcripts, right, you used to be able to, you had a grade, like a grading transcript of all your grades you had in all of your classes. And if you were to apply to college, your high school would have all of your grades on this permanent record, and you would never ask them to send your permanent record out to all the colleges you applied to. Right? They would just make a copy of a transcript and then send that transcript out. Right? So it's the same kind of concept. You're not going to take the DNA, the actual genetic code, and then give it to the ribosomes. Right? You just want to make copies of the parts you need, right? Make copies of the relevant genes, and then send those transcripts out to the ribosome. So then only the relevant proteins are going to be made. So the enzyme that does this is RNA polymerase. So RNA polymerase is able to read the genetic DNA code and rewrite it into a different language, so to speak. It transcribes it into messenger RNA called mRNA. This is occurring right now in all of your cells' nuclei, right? You always need proteins, so you're always going to need to do transcription and translation. So in DNA, right, we know it's double-stranded. One of these strands is going to be used as a template to copy from. And RNA polymerase will be able to make an RNA version, right, of the complementary strand. It's important to note that either strand of the DNA can be a template strand. So RNA polymerase can use this bottom strand as a template for one gene, but it can be using this top strand as a template for a different gene. So genes are kind of interspersed along our chromosomes on both strands of DNA. That's a little confusing sometimes. So transcription occurs in the nucleus, of course, because we're copying DNA, and DNA is stuck in the nucleus. And RNA polymerase is going to use the template DNA and is going to attach RNA nucleotides in a specific sequence to complement the template DNA. At the end of transcription, we're going to get an mRNA copy of the DNA gene, right? Just of that one gene. It's not the entire genome we're copying, like in DNA replication. In transcription, we're just copying one gene at a time. There are 3 steps to transcription. We start with initiation, then elongation, and finally termination. So let's start with step 1, which is initiation. Every gene starts with a promoter. Stop right there, right? Every single gene starts with a promoter sequence. All genes are made of DNA, so every gene starts with a promoter sequence of DNA. There's a region called the TATA box, T-A-T-A. There's like a T-A-T-A that marks the start of every gene. It's part of the promoter. And in order to start transcription, in order to initiate transcription, RNA polymerase must bind to the promoter of this gene. So RNA polymerase recognizes the promoter of a specific gene, and several proteins called transcription factors can help RNA polymerase either bind to the promoter or be repelled from the promoter. So RNA polymerase is going to start copying, or not copy, or is going to really transcribe the DNA message into an RNA message. And again, this starts at the promoter, which is the beginning of every gene. Every gene has its own promoter. This is very important. Make sure you understand what a promoter is. I want to introduce some terminology. The promoter is always upstream of the coding sequence, of the actual gene coding sequence. Upstream means before. Downstream means after. Another way to say that is like upstream means more toward the 5' end. A downstream is farther downstream, meaning like closer to the 3'. So upstream of the coding gene is the promoter, right? And the coding sequence is downstream of the promoter. So this is initiation. RNA polymerase recognizes the promoter sequence, which is the beginning of a gene. RNA polymerase has the ability to open up the DNA double helix on its own. So it opens up, unzips the DNA helix, and it begins to add RNA nucleotides to complement the DNA nucleotides on the template strand. So if the DNA had a C, the RNA polymerase would put a G. If there was an A, right, you would think, oh, A goes with T. But remember, nope, there's no such thing as T in RNA. So RNA polymerase is going to put a U. So it's going to be GU. And then instead of a G, right, if the G is in the template, then the RNA polymerase will put a C, a cytosine RNA nucleotide. Right. So it's going to add in the complementary RNA nucleotide as it reads the DNA template. RNA polymerase can only synthesize mRNA in the 5' to 3' direction. So it can only add new nucleotides to the 3' end of an existing nucleotide. So RNA polymerase is going to then add something else right here. And then right here. And therefore, it's making new RNA 5' to 3', just like DNA polymerase did. So RNA polymerase starts at the 3' end of a template, starts reading toward the 3' end of a template, and then will make new RNA 5' to 3'. And remember that if you're an adenine, you pair with U, with uracil in RNA. Transcription stops in the third step called termination. when RNA polymerase encounters a terminator signal on the gene, it falls off, and the entire mRNA detaches from the DNA. And the DNA double helix can then reform. So what happens here, again, is just the opposite of initiation. At initiation, the RNA polymerase recognized the promoter and started transcription. In termination, it recognizes a terminator and falls off and stops transcription. And now the cell produced an mRNA copy of a gene. So let's watch this video together.
During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
So remember, the DNA template strand is transcribed into mRNA. And the template strand you'll be told which strand is the template strand and which is the non-template strand, also called the coding strand. So a trick is the template strand will always be written 3' to 5'. The coding strand will always be written 5' to 3'. So this is important. If you are shown a template sequence of DNA, 3', T, C, G, C, T, G, 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 5', what would transcription in the nucleus produce? So really take a second and tell me, in your notes, what would the sequence of mRNA be if this template strand were transcribed into mRNA? So pause here. So what you would see is 5', right? If there's a T here, there would be an A in the mRNA, right? This is RNA polymerase doing this. So if it sees a T, it'll pull an A. If it sees a C, it'll put a G. G, C, C, G, T, A, and then C, 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 3'. Never forget the 5' and 3', right? So the mRNA is going to look just like the complementary DNA strand, right? So the coding strand of DNA is going to look just like the mRNA transcript. So again, you might want to pause here to make sure that makes sense to you, because it is a little confusing. But the mRNA transcript, right, is complementary to the template, just like the other DNA strand is. The other DNA strand is also complementary to the template. There's only going to be one difference between the mRNA transcript and the non-template strand of DNA. Can you think about what that is? Right. If there's a T here in the non-template DNA strand, there would be a U in the RNA. But if you see a DNA code that's written 5' to 3', the mRNA transcript would look the same 5' to 3', with the exception of T's would be switched to U's. But in a sequence that's 3' to 5', right? That's template DNA. You can't read a gene that's written 3' to 5'. So that would be transcribed, right? And if, again, 3' T, you would put an A. And then if you see a C, you would put a G. G, you would put a C. And that's how you get this sequence here. And again, we could also call the non-template strand the coding strand because it looks just like the mRNA code after transcription, with the exceptions of the T's and U's. So again, let's try to do this example. I'm telling you which one is the coding strand and which is the template strand. So I want you to tell me the mRNA code. Please pause here and figure that out. All right, so the answer is AUG, CCG, ... And of course, you have to write 5' and 3'. And like I said, the mRNA transcript will look just like the coding strand, or the non-template strand, right, of DNA. That's written 5' to 3', with the exception of T's get switched to U's. Make sure this part makes sense. So pause here and stare at this. Read the book, watch the supplementary animations in the videos. Don't go further until this makes sense. So now we're not quite ready for translation yet because the mRNA has to be processed before it can leave the nucleus. So we're going to go through 2 steps of processing. This happens after transcription, but before translation. Within every gene, there are parts that actually code for amino acids in the proteins, and there are parts that intervene. The parts that get expressed into proteins are called exons. Exons get expressed. Those are the protein coding regions. There are other parts of a gene that intervene, and those are called introns. So, since only the exons are needed, right, to encode a protein, we have to get rid of all the introns before translation could occur. So what happens is introns are spliced out before the mRNA can leave the nucleus. Special RNA enzymes called SNRPs, or small nuclear RNAs, can cut introns out. So these are examples of RNA enzymes or ribozymes. They can splice out. They make little cuts into the introns, cut them out. They form a complex called the spliceosome to do this. That's a really cool thing that they do. So they splice out all the introns that intervene, and they seal together all the exons that actually code for proteins. One more thing has to happen. There has to be a special 5' cap and a 3' tail put on to the transcript. So there's a special cap made of a methylated guanine nucleotide and phosphates. And this helps ribosomes attach to the mRNA transcript during translation. It also helps protect and stabilize the mRNA transcript. There's also a poly A tail. What that means is it's a bunch of adenines, A. It's like 100 or 200 added to the 3' end of a transcript. And this helps provide stability to the mRNA. And the longer the tail, the longer that mRNA will be in the cytoplasm, which means the more protein could be made from it. So if you don't want a lot of protein to be made from a certain mRNA, it might have a very short poly A tail. It won't be as stable. It will be degraded rather quickly, so ribosomes won't be able to continue making protein from it. So we started with the primary RNA transcript, right? RNA polymerase just gave us this, exons and introns. We then had those little SNRPs cut out the introns. We put a 5' cap and a 3' poly A tail. This is now a processed RNA. This is a really good, informative picture that I like, right? This is what a gene looks like. It starts with a promoter, and it has exons and introns, and it has a terminator. RNA polymerase will start at the promoter. RNA polymerase starts at the promoter, and will start transcribing this gene until it reaches the terminator. Then, all the introns get cut out, right? All the introns get removed by RNA processing. A special 5' cap gets put on, and a special 3' poly A tail gets added. And within this gene, we're going to have a start and a stop, as we'll soon see. So you don't have to understand what this part means yet, this UTR stuff, but this is how we're processing mRNA to get ready for translation. So let's watch this video on RNA processing.
The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation.
I just want to add one point, that actually for many years, scientists thought that introns were just wasted space within genes, and that they were quote-unquote just junk DNA. But we now know that introns actually have a lot of functions. So introns actually allow a cell to choose which exons can go into a particular mRNA. And we'll talk more about that in the next chapter. It's called alternative splicing. So it gives us a lot more flexibility in different genes. So from one gene, we can actually get multiple different transcripts because of the introns. Also, some introns help control gene expression by making small RNAs that interfere with either transcription or translation. It's also now recently discovered that introns help crossing over during meiosis. So introns get removed because they're not needed in the cytoplasm, but they might have a lot to do with gene regulation in the nucleus. But regardless, we got rid of the introns, we put on a cap and tail. We're now ready, right? We got rid of the introns, cross those out, put on a cap and tail, and now we put on, we're getting this out through a nuclear pore to find a ribosome, right? We can leave the nucleus and start translation on a ribosome. What gets added before mRNA can leave the nucleus? The answer is D. And pause here. What gets removed? The answer is A. So take a second to fill in 1, 2, 3, 4, 5, and 6. So pause here. So the answers are we're making mRNA, instructions to make specific proteins. Transcription occurs in the nucleus. The enzyme RNA polymerase. Introns have to be removed. 5' cap and a poly A tail, 3' poly A tail have to be added. And here is a video that shows transcription in a more realistic light.
Extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
So let's try this question together. If the DNA template strand had the following sequence, what would the nucleotide sequence of a complementary RNA molecule be produced in transcription? Okay, so pause here. So if this is 3' AGC, AGT, CTT, we have to go 5'. This is going to be RNA polymerase, right? Because we're talking about transcription. So if it sees an A, what is it going to want to put here? It has to be a U. And it sees a G, it'll put a C. T gets an A. C gets a G. C gets an A. T gets an A. So the answer is D. Make sure you could do this type of a problem. So now let's talk about translation. So this is going to discuss how we make proteins after transcription, right? This is going to explain how the DNA code, right, can be translated into an amino acid sequence. Translation takes place on ribosomes in the cytoplasm. We remember from chapters 4 and onward that ribosomes may be in the cytosol, like floating around in the cytoplasm, or on the rough endoplasmic reticulum. And ribosomes can translate information from mRNA into a chain of amino acids. So what we're doing is we're translating one form of information in RNA nucleotides into a different language, right? It's a language of amino acids. So in transcription, we went from DNA to RNA, and then translation takes place outside of the nucleus on ribosomes, right, to make proteins. And every protein is just a unique sequence of amino acids, right? So proteins are what we get from translation. Proteins are made, or I should say amino acids are assembled in an order, just like 5' to 3', but we say these are N' to C'. And that's because the amino end of the first amino acid sticks out, and the carboxylic acid end of the last amino acid sticks out. So we always want to label amino acid sequences with like N' to C'. Just like we label 5' to 3', we do N' to C'. And the 20 amino acids look like this, right? They're just different molecules. And we know that they have different properties. Some are negatively charged, some are positively charged, some are hydrophobic, some are hydrophilic. So the sequence of amino acids, right, determines how they can fold and interact with each other. And how they interact with each other will determine the shape that they form, the structure, and the structure will determine how they can function. So this is where we try to tie everything together. We know that these instructions are found in DNA. But how does a DNA instruction actually encode for proteins? Right, we've been saying this whole class, but how does it actually work? So we know that DNA gets transcribed into mRNA. So mRNA has the same instructions that DNA had. And within mRNA, every 3 letters, every 3 nucleotides of mRNA specifies one amino acid. So every 3 mRNA nucleotides is a code. It's a secret code for one of the 20 amino acids. We call these codons. So the mRNA code specifies the amino acids in a protein. Right? A codon is just 3 consecutive mRNA nucleotides that specify one type of amino acid. So for example, AGC is the first 3 nucleotides. That's the first codon. AGC only specifies the amino acid serine. The second codon, GAC, only specifies the amino acid aspartate, and so on. So this is a way to look at all the different possible codons and how they're divided amongst the 20 amino acids. So you can see a 3-letter abbreviation for each amino acid, and there's a 1-letter abbreviation for each amino acid. So for example, valine is abbreviated with V, and GUA, GUC, GUG, or GUU are the 4 possible mRNA codons that would specify a valine amino acid. There's also a start codon, AUG, that all proteins begin with. Every single protein starts with AUG as the first every single mRNA transcript starts with AUG as the start codon, because translation begins at AUG all the time. But this is annoying. Sorry about that. What this means is that every protein will have a methionine, which is the amino acid that's encoded by AUG. Every protein will start with the methionine first. There's also 3 stop codons. UAA, UAG, and UGA are the 3 codons that will tell the ribosome to stop translation. So ribosomes start at an AUG all the time, and they stop at UAA, UAG, or UGA. Right? There are 61 other codons, or I should say, and if you get rid of the start and these 3, there are 60 other codons that specify one of the amino acids. Right? There's 61 that encode for amino acids and 3 stop codons, so 64 in all. Right? Methionine is always the first amino acid. And the genetic code is redundant and degenerate, meaning that you can have so many possible codons encode for one amino acid. And that's a good thing, because this means even if there's one mutation in DNA, there's still a good chance that that mutant codon would still code for the same amino acid. All right, so that's how mutations don't always have consequences on the final protein, because there's this redundancy. So in order to translate mRNA into proteins, we need to use a codon table. So a codon table matches each codon to its amino acid. So I want you to take a second and look at this codon table. We could read the first base on this column over here, in the left column. So let's look at CAU as a codon. And whenever you're given a codon, we're going to assume that it's written 5' to 3', because mRNA is always written 5' to 3'. So let's find the first letter in this left column, C. Then you find the second base on this top row. So here we go, A. And then we find the third base in the right column. And here we're going to find U. Right, so here's U. And this gives us CAU encodes for the amino acid histidine, which is abbreviated HIS. So CAU is the mRNA codon that encodes for the amino acid histidine. You can also look at it like a wheel, where the first letter is here, then the second letter, and the third letter. But I'm going to give you a codon wheel for the quizzes. And you should use, for the homeworks, just use the one that's in your book. And that's on page 218 of the textbook, figure 12.11. So before we talk about translation in detail, let's review three main classes of RNA that we're going to see play very important roles. So the first type of RNA is mRNA. That's the type of RNA we already seen. So this is the transcript of a DNA code that exits the nucleus and carries the message to ribosomes on how to make a protein. Right, so mRNA has codons of RNA nucleotides that specify an amino acid sequence. The next type of RNA is called rRNA, which is a major component of ribosomes. And the rRNA, along with proteins, make up ribosomes that allow amino acids to be assembled into functional proteins. Finally, tRNA, or transfer RNA. And these are molecules that transfer amino acids to the ribosome so they can be connected to make proteins. So transfer RNAs physically link the message in mRNA, right? It links a codon of mRNA to an amino acid. So transfer RNAs are very important for translating the message of mRNA into amino acids. What type of RNA is made during transcription? Pause here. The answer is mRNA. What type of RNA are ribosomes made of? The answer is rRNA. So ribosomes make proteins. They can be found either on the rough endoplasmic reticulum or floating around in the cytosol. There are two subunits to each ribosome. There is a large subunit and a small subunit. They both consist of RNA nucleotides and proteins. They're mostly rRNA, though, that ribosomal RNA. And rRNA is produced from a DNA template in the nucleolus of the nucleus. That's where ribosomes are made. And the rRNA is packaged with proteins to make the small subunit and the large subunit. The ribosomes will help the three types of RNA interact with each other in order to build a protein in translation. So the rRNA of the ribosome will help the mRNA interact with the tRNA. So all of these RNAs are coming together in order to make proteins. The large ribosomal subunit specifically has ribozyme activity that could create a peptide bond between two adjacent amino acids. And the peptide bond is created many times to produce a polypeptide that can have hundreds and hundreds of amino acids. And it will then fold into a three-dimensional shape to become a protein. Right, so ribosomes are the cell's protein-making factories. Right, and it can be found either floating around in the cytoplasm or associated with the rough endoplasmic reticulum, which is right outside of the nucleus. Right, so a transcript is going to leave the nucleus and then either find the ribosome here on the endoplasmic reticulum, or it could find the ribosome over here in the cytoplasm. A completed ribosome has 3 binding sites. You do not have to know the names of these specifically. I'm not going to test you on those details, but there is an E site for the exit, a P site for where the peptide bond is made, and an A site for where the incoming amino acid comes in. So tRNAs can be in one of 3 positions on the ribosome. And we're going to see how the mRNA kind of snakes through a special channel of the ribosomes. This is the mRNA. So let's zoom in onto the tRNAs that transfer amino acids to the ribosome. tRNAs kind of resemble a flattened 3-leaf clover, and they form hydrogen bonds in specific overlapping locations to give them this structure. There's something very important on this end, which is an amino acid. All right, so tRNA molecules transfer amino acids to ribosomes, and the amino acids are attached to one end. On the other end, there is an anticodon. And an anticodon complements a codon of mRNA. So an anticodon is always written 3' to 5'. So an anticodon is always written 3' to 5', and it complements an mRNA codon. Okay. So, for example, an anticodon on a tRNA will determine which amino acid it can carry. The anticodon, like I said, is always 3' to 5', and it will complement a codon that's in an mRNA transcript. So I want you to tell me right now, what codon would this tRNA recognize? So if the anticodon was GUC, the codon must be CAG. So this tRNA will recognize the CAG of an mRNA, and it will bring along the corresponding amino acid. So what amino acid corresponds to the codon CAG? Well, we need to look at a codon table, right? We need to look at a codon table, and so we said since the anticodon was GUC, the codon must be 5' CAG, 3'. So if we look up CAG on a codon table, right, this is a codon table, so we have to look up the codon. We can't just look up GUC, because that's the anticodon, right? This is a codon table, not an anticodon table. So if you're given an anticodon, you have to figure out what the codon will be. So we figured out that would be CAG. So CAG encodes for the amino acid glutamine. Therefore, this amino acid over here was a glutamine. Every tRNA with the anticodon GUC is carrying a glutamine. When a tRNA has an attached amino acid, it's referred to as a charged amino acid. It's been charged by a very special enzyme. So once the amino acid is attached to the tRNA, it can be used by a ribosome in translation. All right, so again, the codon of mRNA will be recognized by a specific tRNA anticodon. So if the anticodon on tRNA is 3' UGG 5', then what will it bind to on an mRNA transcript? So pause here. And the answer is B, right? The codon would be 5' ACC. Suppose the codon on mRNA is UGG. Then what amino acid does it code for? So pause here. So UGG is tryptophan. UGG encodes for tryptophan. If the anticodon on tRNA is 3' UGG, then what amino acid will it carry? So remember, you can't just look up UGG here, because that's the anticodon is UGG, and this is a codon table. This is not an anticodon table. So we have to figure out what the codon will be, which is going to be 5' A C C 3'. So let's look that up. It's A C C. That's threonine. So the anticodon, I'm trying to draw this is the anticodon of a tRNA molecule. This is, or it's like a 3-leaf clover. And then on top, this is the amino acid that will be threonine. All right, this is the anticodon UGG, and on the mRNA, it'll recognize the codon ACC. And again, I should write 5' and 3'. So that's the key for how translation works, how we're going to basically translate the mRNA language into amino acid language. The anticodon on tRNA is 3' AGA, and what amino acid will it carry? So pause here. The codon would have to be UCU, which would mean it's the amino acid serine, which is the answer D. So there are the same 3 steps to translation. There's initiation, elongation, and termination. This all begins at the ribosome, right? Translation. So initiation is the first step of translation. And the small ribosomal subunit and the first tRNA come together at the start codon of mRNA. So remember, within every mRNA transcript, there's a start codon, AUG. The first AUG marks the beginning of translation. And a special tRNA called an initiator tRNA carrying methionine will recognize the first AUG of an mRNA. What's going to then happen is once the initiator tRNA recognizes the start codon AUG on the mRNA, the large ribosomal subunit will attach and kind of make a sandwich with it, where the mRNA is in the middle, you have the small ribosomal subunit and the large ribosomal subunit. And we have the first amino acid is in the P site. So this is what we're starting with. And this requires energy to happen at every single step. But this is the initiation complex. We have a small ribosomal subunit, mRNA, large ribosomal subunit, and the initiator tRNA in the P site. And that's carrying a methionine to the first AUG. This is the initiation complex, right? This is another way to see it. This is the 5' to 3' sequence of mRNA. The first AUG will be recognized by this tRNA carrying methionine, and then this large ribosomal subunit will attach to it, and this is the initiation complex. So now we're ready for the next step, which is called elongation. And a new tRNA molecule, right? We have the first one is over here. We have the methionine is in the P site. It recognized the first AUG. But now we have the next tRNA molecule is ready to come in and bind to the next site. So this is the A site. This is the P site. And that was the E site. So we had the first initiator tRNA here. The next tRNA will then come in according to whatever these next 3 letters are. So depending on the next 3 letters, the next codon in the mRNA, that will determine the incoming tRNA. So the anticodon on the tRNA will be matched up with the codon on the mRNA, and it will bring over the next amino acid. So you should be able to tell me, actually, if this is carrying leucine, what are 3 possible letters that would be right here that would correspond to leucine? So anyway, once the next tRNA comes in, a peptide bond could be formed between the 2 amino acids. What happens is this methionine gets added to this leucine, and everything slides over. So we're going to see this in an animation. It's a very visual process. It's very hard to explain, especially when I'm not in front of you. So you definitely have to watch videos over and over again to understand translation. But what's happening is the second tRNA will come into the site, and then a peptide bond will be formed between methionine and the next amino acid. And the mRNA will then be pulled down one. So there's going to be, now this site will be exposed. These two are going to move here. So that will leave this thing empty. This is the picture from the book, right? So after, right, consider this is the amino acid chain. We have a new one that comes in at the A site. The entire polypeptide chain will then be attached to the incoming amino acid, right? This is a peptide bond is going to be made. There's that peptide bond made. And then everything is going to slide down one. So this is going to move to the exit site and it's going to be kicked off, right? And this is going to move to the middle. So right over here. And that will leave another spot, right? It will leave the A site open for the incoming amino acid to come in. So one by one, an amino acid, a tRNA carrying an amino acid will come to the A site. A peptide bond will be formed, right, at the P site. And then that amino acid, the tRNA that doesn't have an amino acid on it anymore will then exit from the E site. This will continue happening until a stop codon is reached. So that could be UAA, UGA, or UAG. And when that is reached, a release factor tRNA binds to the ribosome. So it looks like a tRNA, but it's not. It's a special release factor that recognizes the stop codon. And that causes the entire ribosome to release the polypeptide. And it falls apart. The ribosome can now work on another polypeptide. So this ribosome can now find another mRNA transcript and start working on making another protein. This polypeptide, right, which is just the chain of amino acids, will then fold into the proper protein conformation to be functional. In order for proteins to be made in an efficient manner, sometimes multiple ribosomes can attach to a single mRNA transcript and make lots of copies of a protein at once. So this ribosome, this one started. This was the first ribosome that started over here, started reading the mRNA transcript and was building the protein, right? And this ribosome started right after it. This is the most recent ribosome to start on this transcript, but it's going to make its way along, and we're going to get lots and lots of polypeptide chains made from just one mRNA transcript. And there are special proteins called chaperone proteins and the cytosol that will help make sure that the amino acids fold into their proper shape. Right, that will prevent protein aggregation, meaning protein clumping. So let's watch this animation together. This is one of the best animations I could find on translation. Again, your ebook has lots of animations, and I have some more links for you in this slideshow.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation.
So hopefully that was a little more visual of a summary for you, and that could help solidify all the different moving parts. Because like I said, translation is very animated. It's a lot of moving parts at once. So you have to kind of sit in front of a screen and maybe even just rewind it over and over again. And so you understand how the tRNAs are moving, how the ribosome is making peptide bonds. All right, so I really want you to watch some of these videos until everything gets solidified in your brain. So to summarize, transcription occurs in the nucleus, right? DNA in the nucleus served as a template for the mRNA. That mRNA then had to be processed before leaving the nucleus. Once it left the nucleus, right, it had cap and tail, got the introns removed. It then left the nucleus and found a ribosome in the cytoplasm or on the rough endoplasmic reticulum. tRNAs with anticodons carry amino acids to the mRNA at the ribosome. Starting with the start codon, we begin initiation of translation, which is going to bring over tRNAs with amino acids one by one to connect amino acids by peptide bonds. The ribosomes will attach to the rough endoplasmic reticulum if it needs to be as part of the endomembrane system. So this is part that I didn't show you yet, but this ribosome, as it's making these proteins, some of the ribosomes will join and associate with the rough endoplasmic reticulum, and these proteins will be snaked through into these endoplasmic reticulum, and that's where it's going to fold and be modified into its final shape. So sometimes these amino acids are attached together, but they only fold once they're inside the endoplasmic reticulum. Right? So a gene has been expressed, right, when a protein is made and it's operating in the cell. The first few amino acids of a polypeptide act as a signal. It's called a signal peptide. And that has information about where the polypeptide belongs. Does it belong in the endoplasmic reticulum? Does it belong to be secreted outside of the cell? Does it belong in the lysosome? Does it belong in the cytoplasm? Right? The polypeptides can be processed further. So sometimes they need to have sugars, phosphates, or lipids added. And that can happen in the rough endoplasmic reticulum. And the Golgi apparatus also adds some post-translational modifications. We know from the Golgi apparatus that transport vesicles can carry these proteins between organelles and to the plasma membrane as appropriate. So let's watch this video together. This is contained in the ebook as well.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Messenger RNA binds to the small ribosomal subunit, and then the large subunit associates with the small one. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs enter at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which no longer carries an amino acid, leaves the ribosome, and the next transfer RNA, with a complementary anticodon, enters the A site. Each time a new codon sequence on the messenger RNA moves into the A site, a new transfer RNA amino acid can bind. The old transfer RNA paired with the previous codon is passed to the P site. The amino acid it carried is transferred to the growing amino acid chain. As the ribosome moves down the messenger RNA, a stop codon is met. A protein release factor binds to the stop codon and cleaves the polypeptide from the transfer RNA. The ribosomal complex falls apart.
So there is one last video that I want you to watch. The link is shown down here. This is a very, very good video that kind of summarizes everything from gene to protein. Again, this is not going to be enough to just watch it once. I really want you to watch it at least 2 to 3 times. But before we go to that, I just want to point out that many antibiotics work by inhibiting bacterial gene expression. So when you think about antibiotics, right, we're trying to control bacterial infections. What they're doing is stopping bacteria from making their proteins. If they can't make their proteins, or if they can't replicate their DNA, then they can't divide and infect us. So there are certain antibiotics that impact bacterial replication. There are some that directly impact bacterial transcription, and many impact bacterial translation. So things like certain antibiotics you might have heard of, like clindamycin or chloramphenicol, actually just impact on bacterial translation. So they can't make their proteins, but they have no impact on our tRNAs. So human cells are fine to make proteins. Bacterial cells are not fine to make proteins. So I just want to watch this video together now.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Great. So, this is a good kind of sample problem just to make sure you're able to do. If you're given a template DNA code, you should be able to transcribe it into mRNA from 5' to 3', then divide them up into 3s, into codons, and then use an amino acid codon table to figure out the amino acids. So you should be able to do this. Make sure you're able to transcribe and translate the amino acids as shown here. And this is a summary table that I made for to help guide your studying a little bit. So for each transcription, translation, and DNA replication, what are we doing? What's going on? Where is it occurring? How frequently? What are the main enzymes involved? And where does it start and end? You must know this information at the very, very minimum. And I just want to take a second to clarify. I usually don't like teaching replication in the same lecture as transcription and translation because they're not really connected. This particular textbook teaches them all in the same chapter. So that's how I decided to teach it, just to keep the chapters less confusing for you. But in reality, think about transcription and translation as happening all the time as long as you need proteins. Replication of DNA only happens before a cell is ready to divide. So transcription and translation are happening right now in every single one of your cells that's active. But only cells that are about to divide are going to do DNA replication, and that can happen in about an hour or so. So make sure you understand where they stop, start, inputs, outputs, and locations. And that's it. And you'll be tested on all of Chapter 12 on the next quiz, so please study very hard. I know it's a lot of information. And that is the last quiz before the final exam. So that is it for now, and we'll talk about Chapter 13, gene expression, in the next lecture.